अगले गोली Yes, you can start now.
Yes, sir. A very good morning to all of you assembled here in this online platform. We all are gathered here for the second lecture on the lecture series which we are conducting on the theme Indians speaking from abroad. This lecture series is organized from Shri Shivaji Education Society, Amravati's Science College, Congress Nagar, Nagpur, Maharashtra, in collaboration with Microbiologists Society, India. So, Honorable President of Microbiologists Society, India, Honorable Dr. Arvind Deshmukh, sir, Honorable Principal of Science College, Congress Nagar, Nagpur, Honorable Professor Mahendra Dhore, sir, our eminent guest speaker, Mr. Ameya Gupte, sir, from Italy, Dr. Patankar, sir, all the student participants and dear friends. Firstly, I pay my humble homage to our founder president of Shri Shivaji Education Society, Amravati, Dr. Panjabrao alias Biofuel Deshmukh. Now, with the permission of President of Microbiologists Society, India, I would start today's session in which the theme is Indians speaking from abroad. The topic of today's session is genetics and genomics approaches to produce superior yeast strains for second generation bioethanol production. The guest speaker is Mr. Ameya Gupte, sir. Now, before his lecture gets start, I must introduce Mr. Ameya Gupte, sir. Ameya Gupte, sir, is now in Italy. He has in his achievements, he has received the research fellowship in Department of Excellence Scholarship, University of Padova, Italy. He has done his master's degree from Department of Microbiology, R.K. Talreja College, Ulhasnagar, India. He has his publications in some reputed journals in Avishkar in 2012-13, an intercollegiate research paper presentation at University of Mumbai, as well as he has received third prize in national conference, which is organized on the topic recent trends and innovations in microbial world, which is held by CHM College. And the topic was isolation of biofilm forming microorganisms from clinical samples and its inhibiting using actinomycetes. Other participations includes poster presentations in Microbial Diversity 2022 in Italy, then Winter School Metagenomics in University of Padova, Venice, Italy, then poster presentation at CONARC Research Meet under the topic of isolation characterization of mercury resistance bacteria for bioremediation of heavy metal polluted sites, as well as he has participated in one-day conference on wetlands and water management held by Paryavaran Dakshata Manch. Then in NAS 2012 International Conference Urban Lakes Ecology, Rejuvenation, Technology and Economics held by NASS. He has also participated in hands-on training and workshop on molecular biology techniques held at Wilson College, Mumbai. He has his expertise in biotransformation technology, fermentation technology, molecular biology, then resin column handling, chromatographic separation, as well as process development for continuous product harvest and yield enhancement. He has also his expertise in basics of HPLC, GC, GCMS handling and troubleshooting, as well as other microbiological skills. Currently, he is pursuing his PhD in the University of Padova, Italy. He has also served as a scientific officer in Godavari Biorefineries Limited, Koperkhairne, India, as well as R&D Scientist A in Privi Organics India Limited, Koperkhairne, India. He has night duty in charge on Clinipath Laboratory, Ambernath, India. So we are very much fortunate that Mr. Ameya Gupte sir has accepted our invitation to deliver his precious talk in the theme Indians speaking from abroad. So I am requesting you, Ameya sir, that you should please give some details about your journey, that how you reach from India to abroad and what are the steps you have undertaken for it, and then you can please proceed for your precious lecture. Please, sir.
Thank you, Pranita ma'am. First of all, thanks the entire committee who has organized this sessions, especially Science College, Nagpur, Professor Mahendra Dhore, principal of the college, and special thanks to Pranita madam for being available for all my queries and questions. Thanks a lot, President of Microbiologists Society of India, Dr. Arvind Deshmukh sir, for organizing such event. I guess it will be very helpful for the students who are aspiring to pursue their education abroad, as well as to understand the topic of research which are going on across the world. And I will continue with the presentation that I have. So I'll share my screen here. I hope it is visible now.
Yes, sir, it is visible.
Okay. So the topic of today is genetics and genomic approaches to produce superior yeast strains for second generation bioethanol production. Before starting my topic, let me introduce you to my university. University of Padova this year is celebrating 800 years of establishment. So this university is one of the oldest universities in Italy and probably across the world, because the university established in 1222. This year is the 800th year. If you want to have a better understanding of what research we are doing in a group, you can see the logo here, West2 Bioproducts, and just attach www.west2bioproducts.com to know more about our group and the research that we are doing in our group. And before starting the actual presentation, I will try to tell you how you can study or pursue your education in Italy. First of all, for BSc and MSc students. In Italy, you don't have a general courses. The courses are quite specific. So you will have a industrial biotechnology or agricultural microbiology. You can have courses on food technology as well as food sciences. And every course is designed for a specific purpose. So when you decide your courses, when you choose your courses, you have to be very careful what exactly you want to pursue in your courses. And you can change university accordingly, because every university has its own way of, the courses are designed in particular way. For example, food technology. Sorry. Okay, sorry. So every course is designed in a specific way. So be careful while you choose the course and just go through all the details of the courses. Second thing, how you select the university. I will not say just in Italy, but in entire Europe, education is free and you can select the university. Basically, public university I would recommend, because it is government university and it offers a lot of scholarships to you, especially the students who have lower income. And almost in India, considering European standards, we have lower income. So there is a great chance of you to get a scholarship. So basically, what I'm saying is, you don't come here just for study. They pay you for your study, also your fees and your expenses. So it is very nice opportunity to study in entire Europe. Talking about University of Padova, you will be offered a different scholarship. First is Excellence Scholarship, which is merit-based scholarship, and only first ranker of entire course will get this scholarship. Then you have regional scholarships. Regional scholarships is basically on the basis of your income. So Veneto region, where Venetian region, let's say, offers you different scholarships based on your income, and you can prove that you have a lesser income, and you can avail that scholarship. Then there is a departmental scholarship, which is the combination of merit and regional based, means income based scholarships. So these are different scholarships that you can avail depending upon your merit and your economic status. Then the basic documents that you need, this is a list of document. The red colored document that you see are generally for scholarship purpose or your visa purpose. But to get admission in university, you need your curriculum vitae in European style, of course, and transcript of the marks from respective college. You can avail that. Be sure that you should have good marks, because in Italian system, you can get 110 out of 110, even at university level, which is not possible in universities in India. So you have to be at least in good grades to avail the scholarships as well as admissions. Then you also need to write SOP, or a letter of motivation, in which you tell the university or the professors why you are so motivated and why they should admit you in a particular course that you're choosing. Then you have this different documents for application of scholarships. You have to prove your sponsorship. You have to show your property valuation and property income so that you will get the scholarship. Now, next is this PhD, very importantly, and I can guide you better on this because I have been through this entire journey. Before telling you how to get admitted in PhD, I would like to tell you what you need and what is expected from you. Obviously, this stuff expectations is quite big, which is always. But what you need to do is you need to have very good marks. As I said before, that in Italy, at least in Padova University, you can get 110 out of 110. So full marks, which you cannot expect in India, neither in your 10th grade or in your BSc or MSc. So be careful and have good marks as much as you can. So study hard. You cannot take this lightly that you will do everything in master's or PhD. You should start from your early career. Then doing little extra. This is very important, and I will share you a small story. When I was in India, I have done a lot of work during my BSc and MSc. If you see, my publication is in second year or third year of my BSc. And also, while doing my MSc, I have done quite different research that you see, which is presented in Trends in Microbiology. And this was done along with my master's. So you have to be active with your, doing extra things and entire career of your BSc and MSc. More than that, another story is when I was in industry. I have 3 years of industrial experience in which I have changed 2 industries for research and development department. When I joined in an industry, I was hired for enzymological project. But then when I saw there was a trouble in one of our projects, which was supposed to go on pilot scale, it was a contamination, and the person working on that project was a process engineer. According to me, she was pharmacist, and then she got mastered in process engineering. So I saw their techniques and I said to my boss, okay, I can help you in this, isolation, because it was actinomycete, which is growing very slowly, and bacterial contamination grows fast. So it was difficult to separate bacteria from this actinomycete. So we did a lot of things, but doing this extra than what you are supposed to do helps you a lot in entire journey. That gives you a lot of experience. So within 6 months of my joining, which is the probation period, I was handling main project of mine and 2 different side projects. Okay, so this doing little extra attitude is very important to pursue PhD and probably in entire life. You should be very serious in topic because you're going to spend 3 years and probably 8 to 10 hours or 12 hours of entire day on a particular topic that you are selecting. So if you're not serious enough and you're just applying because you see some possibilities to get scholarship and to go abroad, it won't help you. At the end of the day, research is 80% failures and 20% success. So you keep on repeating the experiments, and at the end of the day, you get positive results. So be careful when you select your topic. Be serious on that. And last and very important thing is that you have to be a good salesman. What do you mean by that? So when you propose your research idea to someone, when you want to get admitted in a particular university, you have to sell your idea, basically. You have to sell yourself, and you have to show how you can help this entire group that you are applying to, and as well as how this group will help you. So you have to sell your topic very nicely, and this is what is known as basically your research proposal. Obviously, your idea should have a weightage, but at the same time, along with the novelty, it should be explained in a very nice manner so that people can buy it. And what is expected from you? The expectation from PhD student is to design your experiment. And design doesn't mean that you should write something. But when you're asked some WH type question that why, when, what, so why you're doing this, or what you want to achieve, or what you expect out of it, and when, by what time you will finish this, because time is very important. You have 3 years, at least in Europe, you have 3 years of PhD. So your time is very critical in this sense, because there are just 36 months. So you have to design your experiments very carefully. Then, as I said, you have to be very good salesman, because you have to also help your professors to get fundings, because all your research is based on fundings that you receive from multiple agencies. So you have to give your inputs in writing funding proposals. Then you will be working with multidisciplinary group. So whatever you convey to your group members should be in very nice fashion, very easy way, so that you can talk to everyone. Because as I told you, I'm a fermentation expert, or my expertise in fermentation. But I also work with molecular biologists. I work with statisticians. I work with bioinformaticians. So I have to convey all my results, let's say, to the statistics person we come across in my next when we discuss about my topics. But I have to convey whatever research I have or whatever outputs I got to a statistician so that I can get my work done. I have to inform whatever results I have to a bioinformatician so that he can help me in my work. So you will be working in multidisciplinary group. Then you should be good at disseminating your research work to maybe your group members, maybe to sometimes to undergraduate students, graduate students, or in multiple research forums or conferences. Then doing little extra actually translates into taking initiative. So you have to take initiative if someone is having a trouble with something, and if you have that expertise, you have to go and help him. That way you help your group, and also you get a lot of experience in different fields. Many times here you will get opportunity for reviewing research articles. So you will be referee in one of the peer-reviewed journal, and you will be asked to study a draft or manuscript which is supposed to get published in a particular journal. So you read that journal, you read that particular manuscript, you try to find some flaws in the manuscript, and you suggest some reviews to the journal so that you can make that manuscript very strong in terms of research. So this is additional work you do, but this is quite rewarding because you understand how many lacks we have in while we prepare our manuscript, and that helps you also in your own manuscript, which is the 7th point, wherein you have to produce high quality research articles. probably in high impact factor journal or first quartile research journals of your topic. So these are the things that are expected from you. So you should be very careful while you select your topic and be ready for all these expectations. And to tell you how I got admitted in this PhD, basically, in India, we have very low level of research that we do, because if I see master students here, they are doing a great job. I mean, being a master student, I could not expect such kind of research in my master's. But here they are handling very, obviously they have resources and they are handling very high techniques, molecular biologists, biological techniques, CRISPR-Cas9, etc., etc. So when you are doing your work, when you are doing your master's, try to collaborate with all the possible ways, maybe to industries. If your professor are working with some industries, try to collaborate with industries, try to collaborate with other colleges, so that you will have a group which is multidisciplinary, and that will give you an edge. When I applied, because I had almost 3 years of research experience, I was exposed to quite industrial techniques, that is fermentation or chromatographic separations, and that helps you a lot, because your master's project is kind of very short project. At least in Mumbai University, it's just 6 months project. So you have very less time because you have exams, etc. And I do understand this thing, but it will be my request also to teachers to expose your students to possibly industries and to other research groups, if it is possible. You get admitted in Italy, there is a cycle-based format here. So every year there will be a PhD call from all the universities, approximately in March to May. And then you have to apply to that call. Can please someone turn off its mic? Then there is also a possibility to get admitted during entire academic year if there are excess fundings with professors, but every time there will be a notice published on university sites. So you have to keep on watching the university sites, different universities that you choose, to know if there is any opening, because all the public universities has to produce a notice through which they can hire students. The documents that you need, and these are the documents which are essential while you apply. You need to have European style CV, your passport scanned copy, always ready with you as a identity proof, then transcript of marks, your research proposal, letter of motivation. English proficiency test is institutional based. Every institution has different requirements. Some institutions don't require proficiency test, etc. So just check if you need any. Then you need letter of recommendation, or sometimes referee detail, contact details, and scanned copy of your entire MSc dissertation, because they will evaluate you on the basis of your research proposal. You may not do the work that you are presenting. For example, you may be doing different work than your research proposal, but your research proposal tells the professors your research quotient, how much you can think, what is your novelty, or what novelty you can think of. So put a lot of efforts on your research proposal. It cannot be 1 or 2 day job. When I applied, every day I used to shortlist the professors which belong to my topic or which are doing work on my topic, read their research, and cold email them. Approximately 200 or 300 cold emails I have sent. And finally, I got this opportunity. So you have to be very good salesman. You have to keep on sending your proposals to different professors. And you cannot have the same proposal to all the professors. You have to see what work they are doing. What is, for example, only biofuels can have different small aspects. So you have to check what field they are working in and modify your proposal and letter of motivation accordingly. So now we'll start with the actual topic of my research, which is genetic and genomic approaches to produce superior yeast strains for second generation bioethanol production. And the story is same that you see in this picture. As we have a lot of farm waste getting generated every year, and that you see in from burning this farm waste in Punjab causes a lot of pollution in Delhi. So the idea is to use this farm material, farm waste, than using the food, because food is for humans, and there are a lot of people going hungry bed every day. So it is very necessary to conserve the food for human, and to feed your cars or your transportation vehicles, use the leftover material that is farm wastes. The plan is very simple. We have a pool of Saccharomyces cerevisiae strains in our library, and I have to find the prominent inhibitor tolerant strains, which are now parental strains. I have to check the genetic insights, so genetic makeup of the parental strain. Why, I will tell you, and what all the technologies we are going to use, I will tell you. Then genetically modify those strains to make the CBP strain, consolidated bioprocessing strains. As you know that this Saccharomyces cerevisiae cannot utilize cellulose, which is complex polymer. So we are going to introduce some cellulase enzymes. Basically, there is no one cellulase. There are different group of enzymes which you have to introduce in your Saccharomyces cerevisiae. And then your Saccharomyces cerevisiae should produce its own enzyme for degradation of cellulose, as well as ferment the glucose into ethanol. So everything will happen in one fermentation vessel, and you will save a lot of electricity, because producing a biofuel is technically not feasible economically, because you need multiple vessels to be operated and a lot of electricity, etc. So the aim is to produce a cell which can do everything, which can degrade the cellulose as well as ferment the glucose into ethanol. And then testing this CBP strains for ethanol production. So very simple, but today I'm going to tell you only one part of this entire story, which is this part. And next few minutes, we are going to spend on this small part of this entire story. So this is entire plan, what we have to do. As my topic says, genetic and genomic approaches. So we are going to take the genetics approaches as well as the genomic approaches. Let's say it one by one. First, the screening of yeast cells and next generation sequencing of the parental strains. So we have selected this yeast strains, 4 parental strains out of 16 strains that I found potent for inhibitor tolerance. Now, which inhibitors? Basically, when you degrade cellulose, when you give the pretreatment to the lignocellulosic material, this lignin degrades into many monomers, fumaric acid or ferulic acid or syringildehyde, etc., etc. Also, there are some acids like formic acid and acetic acid, and there are n numbers of acids. So all these acids are toxic for Saccharomyces cerevisiae, due to which the viability of Saccharomyces cerevisiae go down, and ultimately you don't get any fermentation. So the starting point is to find such strains which are tolerant to these inhibitors, and then you can go for fermentation and then genetic modification, etc. So ethanol red is industrial bioethanol strain. So it is from France. Next is TC1517. It is Italian strain from grape mark. Y30 is the South African strain from vine fermentation, and YVGC 13A is Canadian vineyard strain. So we have selected this 4 strain considering different geographical locations and origin, wherein ethanol red is the industrial standard. Just to compare with, we have to have a comparison of industrial standard with our wild type strains. And after this, we need to know that the genetic makeup of our parental strains. These are 4 parental strains now. So we did Illumina and Oxford nanopore technology. So we got sequencing using Illumina technology and Oxford nanopore technology. In Illumina technology, the DNA is chopped in very small pieces and then it is sequenced. So you have a very accurate readings, very accurate sequencing, but very short DNAs. While Oxford nanopore technology needs very long DNA, the longest possible strands of DNA, but it has a little bit more error. Now, unlike bacteria, which has a single chromosome, this Saccharomyces cerevisiae has 16 chromosomes. So if you want to have each chromosome properly sequenced, you need to combine this Illumina, which is short DNA, but very accurate, and the long DNA, which is less accurate, so as to get a very strong sequence, which can go up to chromosomal level. So here I have mentioned a small protocol to have a long DNA, but it is not much needed. While here, this you see half of your phone-sized sequencer, which is pocket sequencer. This sequencer is a mini-ion sequencer from Oxford nanopore technology, and it can sequence at the same time 12 different strains and give you output in 3 days. Very, very, very nice instrument, very pocket-sized instrument, but very efficient. So next thing is sporulation of yeast. Now, as we know, yeast can sporulate, and we are going to take the genomic help of this sporulation ability of yeast. So during sporulation, which is meiosis, there is an event known as crossover event. During this crossover event, due to this crossover event, basically, you get a small variation in genetic makeup. And we want to see if this small variation is sufficient to change the phenotypic characteristics of this Saccharomyces cerevisiae. So first thing is to sporulate the yeast. After a lot of different media, we fine-tune one media, that you see here. And from this media, we spread our yeast culture on this media. Potassium acetate helps this yeast to sporulate, and incubation of this media, this agar plates, is for 30 degrees at 30 degrees for 15 to 21 days. So it's not a simple thing, because once you keep your plates for 15 to 21 days, you know that your plates will be dried up. So you have to take care that your incubator is properly having a particular humidity so that your plates will remain hydrated. But after 15 to 21 days, we get this beautiful picture in which you can see this tetrads, that is 4 spores in ascus, or triads, 3 spores in ascus, or dyads, which is 2 spores in ascus. So we are interested in the spores. Now, next part is to break this ascus and take out every spore, okay? Because we want to see if every spore, when it becomes a colony, has some characteristic change. So we have an instrument known as a Micromanipulator, basically. It's a inverted microscope with 2 microneedles, in which you can, through microscope, you see your tetrad, let's say, and you cut open this tetrad, and by needles, you separate each spore and let it grow to a particular colony. But the thing is that this is very time-consuming process and very laborsome process. And I'm talking about few hundreds of colonies. So this process cannot be used because it is highly time-consuming. So we developed a new scheme, a new process, a new protocol to get maximum spores in minimum time. So basically, I scraped the growth from modified sporulation media that we showed before, that we saw before. And this media, the cells that I obtained, the spores that I obtained from this media, I give the zymolase treatment. Now, zymolase is a commercial enzymes which are helpful to produce yeast protoplast. So it break open the cell wall of yeast. With the help of these glass beads, which are micro glass beads, we make sure that all the cells are properly, or the spores, ascus, are properly broke open. And then we treat this entire spores to 65 degrees for 10 minutes. Now, the rationale behind using temperature here is, even after having 21 days of incubation, you will still see some viable cells. And these viable cells can also grow, which are dormant cells now, which can also grow and produce a colony. We don't want that, because we want a monosporal colony from a single spore. So we want to kill the parental vegetative cells. And by doing this temperature treatment, we found that all the parental strains get killed, or parental vegetative cells get killed. So we have added this extra step. And then after doing this, what you get is a spore suspension. And then you spread this spore suspension at very high density on enriched medium, that is YPD, yeast extrose, peptone dextrose media, to get an enriched colony. So as you see from each cell, each parental strain, I have multiple monosporal colony. MSC is monosporal colony. And out of these colonies, I checked its status, whether it is heterothallic or homothallic. Now, what is homothyllism? Homothyllism is the phenomenon by which a spore or a colony, which is monosporal colony, get diploidized. It becomes a diploid colony by itself. Okay, while heterothallic cells cannot become diploid because they lack the HO gene. This HO gene, homothyllism gene, helps, or what do you say, upregulates the MAT gene, which is responsible for mating and type switching. So we are in need of this homothallic cells. And here the multidisciplinary group helps you. So for this, checking this homothallic gene, I have to do some blast. And obviously, when you have this big chromosomal DNA, which is sequenced, and your own chromosomal DNA, you cannot go on NCBI site, but you want to do it in your computer for your research purpose, because you want to keep that research for yourself. And here your group helps you. Also, you can learn from them. And now I can also help some people in bioinformatics. Then out of these monosporal colonies, I selected randomly 30 monosporal colonies and gave them on YPD. And this YPD media, sorry, I don't know who is it, but, and I don't know even how to remove these arrows, but I'm sorry. Anyways, let's continue with it. Don't consider these arrows, they are not from me. Then I use the Python tool. We grew this monosporal colonies for a growth curve in 96 well plate, so that I can take reading after every 5 minutes. Obviously, we have a analyzer to do that. Then I use the Python tool, which is Python growth curves. And using this growth curves tool, we selected the parameters like growth rate, T max, which is time max at which there is a maximum growth rate, and lag time. And using these parameters, we produced this PCOA, that is principal component analysis charts. And as you can see, in that 30, as you can see, this 30 monosporal colonies, along with parental colonies here, if you can see this PAR, which is parental strains as a control, you see some groups and some outliers like this 7th colony or 57 colony. So we selected, just to narrow down, 1 or 2 colonies from each group and process it further. Now, next thing is to understand if there is any change in phenotypic nature. So we put this all the selected 12 colonies in stress conditions. Now, the selection of stress is like this. For example, consider the origin of our parental strains. Ethanol red is a industrial strain for bioethanol production. When you say bioethanol production, there is a lot of salts involved into the fermentation. So NaCl is considered as one of the stressing agent. Or if you consider YVGC13A, which is Canadian vineyard strain. In Canadian vineyards, before few years, they were using copper-based pesticides. So there is a lot of copper still in the soil of vineyard. So copper is considered as a stressing agent for this particular strain. And this MTC is maximum tolerant concentration. So we give the maximum stress to those parental strains as well as the monosporal colonies to see if there is any phenotypic change in terms of growth curve. And then we did analysis of the growth curve. As you can see from this result, these are just two strains that I have shown, ethanol red and TC1517. When we give a stress of 7.75 grams per liter or 7.5% of NaCl, there's a huge change in the growth curves. The magenta line that you see is the parental strain for a comparison. The red one is worst performing strain, while the green one is the best performing strain in terms of growth. And here we can conclude that when you have monosporal colonies, due to this crossover event during meiosis, it generates a phenotypic change, and that phenotypic change is sufficient to see visually under stress conditions. So they basically respond to the strains differently. Also in terms of glucose, because glucose is one of the nutrient, you don't see much changes. But when you again plot the PCOA chart, you can easily make out because software gives you different readings and you see very dispersed, monosporal colonies or different readings on your PCOA chart. And by this method, by again forming a PCOA chart of this, we cut down to the 6 strains just to narrow down. And we want a further confirmation by metabolic studies. Now, our group is in collaboration with the Perugia University in Italy, and they have a very strong group which studies this yeast, and they have developed a way, as you know, this FTIR, Fourier transformed infrared spectroscopy, is the fingerprint for every chemical. Just like we have a human fingerprint, which is unique to a particular human, this FTIR is unique to a particular chemical. So this Corte et al., as you can refer whenever you want, has developed a method by which they want a metabolomic fingerprint of every strain of Saccharomyces cerevisiae so that you can recognize Saccharomyces cerevisiae using that fingerprint. So we went for this metabolic analysis. As you can see, these are the heat maps of different FTIR spectra that we have. And we are trying to compare this heat map with our growth curves. Right now, we have not, analyzed this data because we are in holidays right now. Probably from 1st September, we will start again. But this is just to give you an idea how we are going to analyze this metabolomic results with our growth curve results. And probably this will help us to make our conclusions stronger that there is a phenotypic change also in terms of metabolomics, not in just terms of growth curves, but also in terms of metabolomic analysis. And so this was entire story. These are a few references that you can refer. And thank you for your attention.
Thank you so much, sir, for your valuable guidance. Really, you have emphasized on the topic regarding bioethanol and biofuel production, as well as each and every step how we have to perform the experimentation on that. So hopefully all the participants have got it. And also you have elaborated your journey from India to abroad. What are the steps should be taken for something to achieve? That is emphasized from your side, because most of this times students are unaware about the things. What we have to means present. So presentation is very much important. That if your topic is liked by the other people and if it is accepted by the people, then you can achieve that. So if you have just told that you have sent more than 200 to 300 emails, and then you have got this opportunity, and every time you have submitted some different proposal, not the same proposal to all the personals, so that you have
Yes.
taken lots of efforts for that. That efforts are
So-
important.
I will tell you also one thing, that the people probably say that they don't have time, but understand that I was working from 9 to 6 in a company. And considering the Mumbai travel, you have to understand that 4 hours more in the travel. So you are free after 10 o'clock, after
Okay.
your dinner, etc., etc. And then you start searching for different professors, reading their articles, and start making your own proposals.
Yes.
So every day you spend from 10 o'clock till 12 o'clock, 1 o'clock, okay,
Yes.
to make a proposal. You make proposal after maybe a week, because it's not easy to make proposal. You have to read a lot of things, because you want to show that you are well aware of this topic. You cannot just
Yes.
write very randomly anything. So because this is the only opportunity that you have to impress the person, because the person only knows what you have written, and
Yes.
he is going to judge you on your writing. So you spend
Definitely.
a lot of time every day, and you cannot say that there is no time, because you want it. So students who say that, okay, they have this college, they have this thing, that thing, no, you have to work if you want to get there.
Yes, sir, definitely. This is very good suggestion from your side, that yes, this is the fact that students and people think that they don't have the time to do such things. But whatever is important for our career point of view, that should be done by the students itself. So accordingly, if the efforts have been taken, then we can achieve our goal. So now in the chat box there are some questions or queries, sir, for you. So I'll just read it.
काय दर्शन काय बोलतोय नीट बोला.
For audience, if you have any queries, you can ask also.
क्या हुआ?
Before that, madam.
क्या हुआ ना, मैं सुबह 2 बाल्टी लेके गया, मैंने पानी भरा, लेकिन एक बाल्टी यूज नहीं हुई, फिर मैंने रख दी, मैं इतना पानी
कामं करतो, मला फोन मी नेहमी
Hello.
खिशात नाही ठेवत ना.
Second, one
मी
second.
दुसरीकडे
There is noise.
जायचं असतं का मला.
Can you mute a person which is not asking questions, please?
Deshmukh, sir. Yes, there is some noise.
Hello.
Yes, sir. Please, sir.
I have removed Vivek Parab.
Or maybe we can unmute or mute all the participants and only unmute
Yeah,
the
The
participants which
problem is
are
that
asking questions.
problem is that many participants do not obey the ethics. They are keeping their mic open. It is a very wrong, very wrong.
No, I think that it is not purposefully, but yeah, anyways, let's continue.
Correct. So I have removed 2-3 participants today. So again, I request other participants take precaution.
So do we
Okay,
have
you
any
can
questions?
proceed. You can proceed for answering the questions, please.
Yes, sir. Participants, if you have any queries, please ask, sir. Or please, you can put it into the chat box.
In the chat box, there is one question. What is FTIR?
Yeah, what is FTIR? FTIR is Fourier transformed infrared spectroscopy. Basically, it's by the help of infrared, we try to check the bond, the tension between the bond, and by which we understand if a particular molecule is if it is lying between carbohydrates or proteins, or if it is alcohol or acid, etc. Basically, the functional group of a particular compound. So it is the way by which we have some idea, basically, to categorize a particular metabolomics into different components. If it is acid, carboxylic acid, alcohol, or carbohydrate.
There is one more question in chat box. Please go in chat
Yes,
box.
hemothalic and heterothalic means what?
So homothalism is basically a characteristic of yeast to get self-deployed. This homothalic gene helps I'm missing the word. It combines two spores and generate the diploid cells. It gets self-deployed, while the heterothalic cells cannot get deployed itself, and they become haploid cells. Now, if you consider this ER, which is ethanol red, now ethanol red is an industrial strain, and it has to be very stable. While the wild type cells can have this possibility of having variations. Being an industrial strain, it has to be stable strain so that every time you get the same productivity, same concentration of ethanol. So those strains are particularly designed in the way that they knock out the HO gene, homothalic gene, so that there will not be any variations in the genetic makeup of those industrial strains. While wild type strains, probably many times they have this HO gene activated, so they get self-deployed.
Okay. So one
Dr.
more question
Ameya?
is there. What
Dr.
is
Ameya,
Yes.
can I talk? Yes.
yes, ma'am.
If-
You can. Yes.
Actually, this is not a question, sort of. First, I will appreciate your talk on the strain improvement zone, which was your research topic, and which is applied to the industry, and the protocol which you are following there. As from the research, there should be project return, there should be consultancies, there should be fundraising. That's very good. But one comment I would like to know, which you have stated, that India has low standard of research. As all Indians are listening, I would like to throw some light on it. So what is your opinion regarding it?
Yes, I do understand that research is very, money consuming, very
Yes.
money consuming, basically,
Right.
process. And we don't have money for basic research where we focus more on applied research. I do understand that. And when I compare my stay in India and here, it's a huge difference. But
why
we have to find,
it should be like in India, why it is there?
It's all about money.
It's all about money. But why
It's all about money. No,
consultancies are less in India? Because see, all children are listening who are doing research, who want to do research later on. So I accept your and appreciate your writing n number of proposals. You being, when you was an Indian, you could write so many proposals. That means they have brain, but there are certain hurdles in India, because once we have to write the proposal should be society benefited, they should be fundraised, and the proposal should be in such a way that it should require very less instrument facility, like you mentioned, your pocket sequencer. It must be very costly. It is not possible in India. Yes. So these are many.
I'm just telling you, even the one cell of that pocket sequencer is 800 euros.
But at university level, these researches are considered, or an in research institute, where actually we have to follow some protocol of the university. Ours is not the only one subject which is very specialized, like zoology, botany, chemistry, and all. We have to follow, and we get that much of only funding. Like in our PhDs also, we sanction one project on the basis of our experimental findings. So it's always good to write the proposals, and as you could write many proposals when you was Indian, our all children can write, but they should understand that there are hurdles in our country to raise the funds for research, and that is why it is lacking. This is what I wanted to say. Are you agree with that?
Yes, yes, ma'am. I do agree with the fundings that we have with our
Yes.
funding agencies also, and there is no scope. I'm very sure that there is no much scope to
According
have
to
a
me,
basic.
yes, Nobel Prize to the researcher to be given to in very less economic way, finding the very applied things in the society, that should be the Nobel research. And all you are doing is very, I mean, it is applied at pilot scale. It's very good, strain improvement. But there are n number of questions in my mind. The scale up is there, whether stability is there, whether tolerancy is there. So that I will discuss on your mail. So this was my point. Ki children should not think that India is not boosting the children. They have the capacity. As you have written 250 proposals, they also can write, and they also can go to molecular biology research in abroad country. Okay.
Yes, ma'am. Actually, I would like to say, and basically in tune with whatever you said, basically we have a lot of potential. I do agree, because maybe in future you will see many Indians talking to you
Yes,
in this
of
particular
course.
series.
Hmm.
But the problem is also funding. We don't have funds for basic research. We want applied research. That is true. But there is also, there are institutes which I know. For example, we were in collaboration when I was in industry. This industry was in collaboration with Institute of Chemical Technology. Okay. And okay, it's a well established institution. It has a lot of funding, etc. But all of that funding come from private players. They are very less dependent upon government institutions. For example, you can have a small collaboration, like teachers can have a small collaboration with a small industry, which will help them to do something.
Yes, yes.
And by this method, you can get money. I'm not saying you will get a lot of money and you can do purchase a sequencer or something. But
We are doing
if you have, because I find there is no bridge between academics and industrial. If
yes,
you
there
can
is
generate a
there is the
small
connection.
bridge
There should
and
be.
send the kids to do a small work in industry, but they will be exposed to the industrial environment, the SOPs, and
Yes.
obviously industry has a lot of things that obviously academics cannot afford, because all the HPLC, GC, the small parts cost you a few thousand rupees. But this is the way, means as far as possible, please try to send your students to industries or the research institutions. Try to collaborate with them. Obviously, it is not possible, also considering the syllabus that we have very less time for the master's project. But there is a lot of potential
Yes.
that even students can think of. Means it's not just, means your syllabus
That
or your
children will do.
university should not stop you from doing or writing anything. Okay, you can always apply abroad, but you can also try to collaborate the way possible.
Hello.
Dr. Gupte,
Yes, Professor.
Hello.
please,
Yes, Professor.
please write your mail in the chat box.
Sure.
May I make?
Hello. Write your mail in the chat box.
Yeah.
So
Hello.
further people can do the inquiry on your mail.
Hello.
Yes.
And
here is my
yes.
here is my simple email, Ameya for microbiology@gmail.com.
Okay.
The interested people can reach me on this email. No problem.
And after one or two questions, now we will conclude. One or two questions.
Hello. Hello.
Yes.
Ameya, you have presented very well. And
Thank you.
in the initial speaking, what you have shown, what is needed by the student, and what are the expectations, that is very, very true. Here, our students sometimes they are lacking. That must be accepted. And another thing is that what you have added at the last, that you must go to the industry. So that is very true, and that can be the possible way. That is very important. Even not only the big institutes can collaborate, but this is my experience. I am now retired person. I worked in a very remote area, that is at Ballarpur, which is in the district Chandrapur, and there I spent 27 years, where I found out the problems of industry, and nearly in the years of 2000, I consulted the industries by generating the fund of nearly about 5 lakhs. So this is possible, and it was my undergraduate college. So you are very true that we must go to the industry. We have to come out of the our colleges and go to the industry, find out the problem, and give your expertise. So thank you for nice presentation.
Yeah, this is the story. Basically, teachers who have a lot of experience should maybe shortlist a small institutions, small industries, and give them some input that, okay, if you're doing this thing, we can take care of this part. Let's say just the what do you say, industrial waste that they generate. Just write a small proposal for industry that you will get benefited if we take care of this part. Because I know that there are master students who are degrading, you're doing on biodegradation work, etc., etc. Because according to me, all the industrial industries have problem of this ETP.
Yeah.
Okay. If you can take care of their ETP, they will be more than happy.
Sure. That is there.
So a small thing can generate a revenue. Firstly, for the department, you can improve your infrastructure. But more than that, the students will get opportunity to visit industries, talk to industrial people. Firstly, they will get job easily, which is also one of the thing for many students. And secondly, they will, or rather teachers will have funding to go for more research.
Yeah, sure. That is exactly what is now it is expected and what we should do. So I know some industries from Mumbai in Padova area, which are the fisheries packaging industries, and they want certain type of the enzymes. They are searching for the colleges who can give the production of that enzyme, but the colleges are not coming forward. So that is the story in the Mumbai, which I know at present.
Okay, because there are many industries who want a cheap labor, basically. Because if industry hire a person, they have to pay all the structures that you have to follow by according to law. But if you have college, they can probably give 5,000 rupees to a person and make him work as a slave, obviously. But by that way, students get an opportunity, teachers get an opportunity. And I have seen many ICT professors, Institute of Chemical Technology, IIT professors, who are consultants in many industries. Means we used to have in Godavari Biorefineries Limited, we used to have consultant from ICT. Even in Privi, we used to have consultant from ICT. So all these professors who can help industries can be a consultant, and probably by this way you can collaborate.
And now there are funding industries, funding institutions also. That is BIRAG, BIG, and then the other things are there. But what happened? The projects which are coming, they are not giving the proof of concept, and the ideation is not proper. So that problem is there.
Yes.
Gupte sir, it is a kind request that please write your email in the chat box because
I have written many times actually.
Sir, but it is
No,
not
you can't
visible.
Oh, sorry. Okay. No. Sorry.
Yes,
I guess it was for a particular person. I'm sorry.
Okay. So because participants can ask their queries on your email too.
Madam, I have shared his email ID.
Sorry. It was for
Yes.
Shikha ma'am.
Okay.
Yes.
Now
yes,
it is for
absolutely.
everyone. Sorry, I'm
Yes.
very sorry.
So there is a
Okay.
one question
And,
from my side in the chat box, sir.
Sir, there
Can
is
you
a question
please-
that what is the
can you please
mutation rate
ask
of
questions?
yes sir, what is the mutation rate of yeast cells if we are growing a wild organism at optimum condition?
Well, they are quite stable They are quite stable. I don't know the point mutation if there is any, because right now we are not studying the point mutations, but they are quite stable. Only thing is that when they come across a particular stress conditions, yes, there is a possibility of mutation, but otherwise, yeah, they are quite stable. Yeasts, Saccharomyces cerevisiae especially.
Thank you for
Okay.
answering, sir.
And one more question is there, sir, that what is the duration of the project with its quality, and finally the funds. So that is one of the query from one of the participant. The prime
So,
problem for
yes.
PhD in India is like that. Yes.
No, look, the thing is, yes, there is a difference between approach. For example, when I was working in India, there was a different approach, and here you have a different approach of research altogether. There is possibility. I mean, I do understand that 3 years is nothing because I'm suffering quite a backlog also due to COVID, etc. Many instruments were not working, or you were not supposed to go, etc., etc. But yes, if you have more time, obviously you can do more research, but research is something that you cannot finish. I mean, I don't think you can finish the research. Till the time you are in your course, you will keep on doing something, and it is never finishing tasks. So you have to, make a full stop somewhere. And 3 years is not a huge time, but it is not even less time, I would say. So you have to design your project accordingly. You cannot say that the project which will go for 10 years, I will finish it in 3 years. It is not possible, because understand, we are microbiologists, so our organism takes time to grow. You cannot grow an organism in 2 hours. If it is taking 48 hours, it will take 48 hours. And till the time, you cannot go ahead. Maybe you can do some other experiment, but you cannot conclude a particular experiment. So yes,
Okay.
duration is the matter.
Yes, sir.
And funding,
So thank you.
yes, because more the fundings you have, more sophisticated instruments you have. Now I have shown you 30 colonies which are done in triplicates, their growth curve. Obviously, I need a machine which will take reading for me 24/7. I cannot sit there to take reading, that to in triplicate for 30 strains in 3 concentrations. So yes, more money is better. It saves a lot of your time, a lot of your energy and efforts, but you have to find a way. Means there is no other way. I
Yes,
see.
sir. Yes, sir, definitely. And sir, there are some more questions, but it is a kind request to all the participants that Dr. Gupte sir has just shared his email ID, and on that email ID you can feel free or comfortable to ask him the queries. So thank you so much all the participants that you have patiently listened Ameya Gupte sir, and thank you so much Gupte sir, that you have very well presented your topic and also solved the queries of the participants. Thank you so much to
Thanks a lot, madam, for inviting me here.
Yes, sir. Thank you so much, President of Microbiologists Society India, Honorable Dr. Deshmukh sir, as well as a National Coordinator of MSI, Honorable Dr. Patankar sir, as well as Honorable Principal of Science College, Congress Nagar, Nagpur, Professor Mahendra Dhore sir, for giving this opportunity to conduct this program. And once again, thank you to Deshmukh sir for such a nice idea on the theme that we have to conduct the lecture series on the Indians speaking from abroad. So it may be or it must be fruitful to all the participants. So thank you so much. Thank you, Gupte sir.
Thanks a lot.
Thank you.